All right, hey everybody. We're going to take a look at Chapter 5. We're going to start with Part 1. And with Chapter 5, we're going to be looking at eukaryotic organisms. So we've looked at the prokaryotes in Chapter 5. And so now we're going to take a closer look in detail at the eukaryotes. So we kind of went over the differences in Chapter 1, and now we're going to go into more detail here in Chapter 5. Okay, so a eukaryotic cell, starting out, is defined as a cell that differs from a prokaryotic cell chiefly by having a nuclear membrane. We'll talk about that. And membrane-bound subcellular organelles. And then this last one here we're not going to focus on as much, but it is part of the definition. It's called mitotic cell division. If you've taken general biology, you studied mitosis and meiosis, and that's in reference to mitosis. But we're really going to focus on the first two, the membrane and the membrane-bound organelles, or the membrane-bound subcellular organelles as they're called here. So what I want you to know about eukaryotes starting out, less about the technical definition. I'm not going to make you recite that. I'm not really worried that you can recite this in its exact sentencing here. But what I want you to focus on is the two differences as they're stated here. So there's lots and lots of eukaryotes, and there's lots and lots of prokaryotes, and they can all be very, very different. Eukaryotes are all very different from each other, and prokaryotes can all be very different. But from a very generalized perspective of what are the general things that we see as a fundamental difference, we see that in eukaryotes, their DNA is always surrounded by a membrane. And we refer to this membrane as a quote-unquote true nucleus. And then here they define it as a well-defined nucleus, but we like to call it a true nucleus. In addition to that, they have internal levels of organization called cellular organelles. The term actually derives from the sense that they are similar to organs of our body. So if you think of humans and want an analogy, or just any animal really, but higher level animals like humans, if you want to kind of make an analogy between humans and eukaryotic cells, just to kind of give us a perspective, if we think of ourself as the cell, organelles would be kind of like organs. And so the idea is that within a cell you have these distinct little components that all serve their own function to the overall whole, kind of like our organs give us functions that serve the overall whole, right? Our liver does one thing, our kidneys do another, our heart does another. It's kind of like how organelles function in a cell. In prokaryotes, we don't really see this. We see kind of one cell in which everything takes place within the confines of that cell. We don't see distinct little units that all serve a better purpose. So there's a very, very dramatic difference between these two cell types, and it really comes down to the organization within the cell. Now, when we talk about eukaryotes, these are the organisms that you're most familiar with, whether you realize that they're a eukaryote or not. All plants, all animals, fungi, and then protists, which we've talked about a little bit, are the algae and protozoa. These also are eukaryotes. So if you can see it with the naked eye, it's a eukaryote. And these are typically the plants and the animals and a lot of fungi. Protists are single celled, as we'll describe down here, but anything you can see with the naked eye is a eukaryote. And this line here, as it states right here, but this line here attributes to that is that they can come in both single and multicellular form. So when we see it with the naked eye, that means it's a multicellular form. Individual cells are too small to be seen with the naked eye. So the only time you actually see an organism with the naked eye is when you actually have many cells grouped together, meaning you have a multicellular organism. And that only exists in a eukaryotic form. There are no multicellular prokaryotes. You do have colonies and you do have biofilms that become visible, but they still operate as individual cells within the whole. Now, it is true that some eukaryotes are microscopic. So if you see, or if you're looking at something under the microscope, it could either be a prokaryote or a eukaryote. And we'll describe more differences, but microscopic could be both. Something macroscopic, visible with the naked eye, has to be a eukaryote. There maybe is one exception in the whole world that I'm aware of, but you know, this is pretty much true regardless of the example. Okay, so, and just to clarify one more thing, the protista are defined as the single-celled eukaryotic organisms. They are the more common single-cell or microscopic forms of eukaryote. Fungi, as we described, can come in both micro and macroscopic forms. So they can be either, and we'll talk about more of those in the second part of Chapter 5. A few more differences here. on the microscopic level, a eukaryotic cell typically is 10 times bigger than a prokaryotic cell on average. So even though on a microscopic level they're both very small to us, anything that we can't see with the naked eye generally is considered microscopic. On that level, though, there's a difference at size when it comes to the microscopic comparison. So they're about 10 times bigger than a prokaryote, eukaryotes that is. And we also see that they have more advanced features that we'll get into with this first section of Chapter 5. We'll talk about a cytoskeleton. This is a feature that really allows cells to group together and is really a necessary feature for a multicellular organism to form. So we'll talk about that. We see some advanced motility features. We'll look at flagella in a eukaryotic form, and we'll see how they're similar but much more advanced. And then we'll talk about the membrane-bound organelles I mentioned, plus some other things. So these are just a few things in reference to what we're getting ready to talk about here. Okay, so one of the things I want to do is just have you make a quick drawing to compare the difference between a eukaryote and a prokaryote. So let's go, I'm going to go here to my paint document. You can pull out a scrap piece of paper or do it right there in your lecture guide. But what we want to look at here is just a basic stick-figure drawing between a eukaryote and a prokaryote, and then focus on things like the size and the internal structures that we would find in just the eukaryotes, and then those that we would find in both. And generally speaking, if we find it in a prokaryote, we'll also find it in a eukaryote. However, if we find it in a eukaryote, we may not necessarily find it in a prokaryote. Hopefully that makes sense. Okay, let me start with my labels. Over here I'm going to do my eukaryote, and over here I'm going to do my prokaryote. Now, when I draw a eukaryote, I'm going to draw it significantly larger than my prokaryote. And this could be any shape. I'm just kind of going with the general box type shape here. That's about the size comparison. About a 10 to 1. That may not quite be 10 to 1. But that's kind of the general difference is fairly significant just upon a basic observation. You would immediately realize, oh wow, that's significantly bigger. So whether it's exactly 10 to 1 or not isn't important as long as you recognize one is much bigger. Now, going back to our list here, how much larger? 10 to 1. Now, what internal structures would we find in both cell types? So, one, we're going to find a cell membrane. And a cell membrane is a part of the cell that separates the internal and the outside world, the internal cell and the outside world. So a cell membrane is a feature that we will find in both types of organisms. And in fact, a cell membrane is really one of the defining features of all life forms. So a cell membrane is something that we would find in both. And you know what, let me just scoot this over a little bit. What I'm going to do is I'm going to put my features common to both here in the middle, and then I'll put only those on the eukaryotic side over here. So a cell membrane would be found in both types of organisms. We'll label this as common to both. Alright. Just scoot this over a little bit more. Oh, sorry. I know this is painful to watch. I apologize. Well, how about we just trash that? Right, I'm going to get it together here. Common to both. Okay, thanks for being patient. Alright, so another feature that is common to both, and actually let me just draw a line, one more thing here to clarify what we're talking about. Cell membrane, cell membrane. Another feature that is common to both are these units called ribosomes. And we talked about ribosomes back in Chapter 4, and we mentioned that these are structures that synthesize protein. Okay, now I'm going to draw a line to these here in a minute when I add some more features to the eukaryote. So for now, I'll leave those blank because they're structured a little differently. They're found in both, but there's a difference as to kind of where they're found. So for now, I'll leave them blank over here in the eukaryote. In a prokaryote, they're more or less just floating around the cell kind of anywhere within this internal space. And they don't, from our vantage point here, they wouldn't look much more than anything like little specks. So those are the ribosomes. Another feature that we would find in both is what's called the cell cytoplasm. Cytoplasm, this is going to be tough to point to because cytoplasm is the internal fluid of the cell. So this is just pretty much all the liquidy stuff, so to speak, within the internal compartment of the cell. So in a eukaryote, we'll find that just anywhere within the inside, and the same is true of a prokaryote. So the cytoplasm would just be the liquid inside. And then one more feature that we're going to talk about that would be common to both would be the genetic information. And by genetic information, we mean we need DNA and RNA. So let's clarify that here. Genetic information involves DNA and RNA. Okay. And I'll draw a line to that in the eukaryote here in just a second. And for now, let me go ahead and fill this in. In a prokaryote, the DNA is this big kind of long strand that's kind of coiled up in one part of the cell over here, kind of like that. So we would point a line to that over here like that. Okay. Now let's go back to the eukaryote and let's focus on some features that are common or that are unique just to the eukaryote. And then we'll kind of connect some of these lines that I haven't drawn over here. So one feature that we find in a eukaryote that we don't find in a prokaryote, as we mentioned earlier, is the nucleus. And this is where I said earlier we could call this a quote-unquote true nucleus. And I don't know that it's really necessary to get into why they call it that. That would take time, and it doesn't really help, I don't think. So there's a reason they call it a quote-unquote true nucleus, but for now, just recognize that that's the term. What this really means is that we have our DNA within the cell separated by its own membrane. So let me go ahead and draw the DNA here. So here's our DNA, kind of like we saw in the prokaryote, all kind of coiled tightly. But the difference is we actually have a membrane surrounding it that separates it from the rest of the cell. So if you look over here at the prokaryote, the DNA is just kind of sitting within the membrane, floating around the cytoplasm. Over here in the eukaryote, our DNA is separated by this membrane. So here's the DNA itself inside the nucleus, and then here is the nucleus itself, this membrane around it. Okay, you might think, well, who cares? What's the difference? Well, you can kind of think of like the DNA of the cell as kind of like the brain of a human. It's more or less the most important part, and it also is the central area where things generate from. Kind of like everything you do originates in your brain one way or another. I mean, you could probably argue that, but you know what I mean. The nucleus is similar in that everything kind of generates from the nucleus outward. Okay, so it's kind of like the brain of the cell. So having more separation and having more protection is pretty significant, and it's kind of like the better off, more protection your brain has, the more advanced features your brain has, the more well off you're going to be as a species as a whole. Kind of similar in that sense. So that's a pretty big deal. Another feature that is common, or that is unique just to eukaryotes, is a part of the cell called Endoplasmic Reticulum. And this is often abbreviated as ER. Now, here in a bit, we'll talk about there's actually two different types. But for now, I'm going to kind of keep this somewhat simple and move on here pretty quick. Endoplasmic reticulum branches off from the nucleus, and it's kind of this folded membrane-like structure that branches off usually from kind of one side of the nucleus like that. It can't actually surround it in certain cells, but we'll just kind of keep it simple. And you can see this kind of irregular shaped membrane branching off the nucleus. And that, in a very simplified explanation, is the endoplasmic reticulum. Now, in one part of this is where we will find ribosomes embedded within the endoplasmic reticulum. And here in a bit, we'll describe this as being what's called the rough endoplasmic reticulum. If you took general biology, you may already be familiar with that. But at least in one part of this, we find the ribosomes that we mentioned earlier embedded within here. So here's where the ribosomes are located. And that's kind of why I waited to draw that arrow because they are specifically within that area. There are some that can be floating around the cell, but this is where most of them are found, and it's kind of the main center for the ribosomes in the cell. So you see here already a difference, whereas in a prokaryote, DNA is floating, ribosomes are floating, everything is just kind of scattered. In a eukaryote, we see structure. DNA is housed within the nucleus, ribosomes are structured within the endoplasmic reticulum, and we see a more advanced organization, and that's pretty significant. Let's go for a few more features here. Another one that is unique only to eukaryotes is called a Golgi apparatus. Golgi apparatus is another folded membrane-like structure that's going to be found separate from the endoplasmic reticulum. So it's usually going to be located just off to the side of that. Make that a little thinner. And it usually consists of a series of flattened sacs kind of stacked on top of each other, kind of like this. And that is called the Golgi apparatus. We'll talk about what that does here in a bit. For now, we'll just keep it to the drawing. Couple more that's found in all eukaryotic cells. We'll just do one more, and that is the mitochondria. Mitochondria, you may be familiar with, often referred to as the powerhouse of the cell. We'll talk about why that is. But the powerhouse of the cell generates energy, and mitochondria often have kind of a bean-looking shape to them. Maybe kind of like this. And there can be many of them floating around the cell. Doesn't have to be just one. So I'll just draw a couple there. And these are the mitochondria here. Alright. So when we look at a eukaryotic cell versus a prokaryotic cell, we automatically see a significant difference in terms of the internal organization. Over here we see lots and lots of structures built within the cell, and these collectively, by the way, are referred to as organelles. Let me just make that label over here. These all can be referred to collectively as cellular organelles, or we'll just call them organelles. So these are kind of like the quote-unquote organs of the cell. They have a structure, and they help the cell achieve a higher level of functionality. Over here on the prokaryotic side, we see everything just kind of loosely scattered about in the cell. We don't see a lot of organization. And so what we end up finding is that the difference translates to a higher level of functionality on the macroscopic level. I should say both the micro and the macroscopic level. Okay, so when we look at how multicellular eukaryotes come about, one of the main reasons is because the internal organization allows for a greater level of macroscopic organization. Okay. So going back and looking at that list here, you should be able to answer these questions here. Now, we didn't cover all of them, but we'll come back to some of the other ones here, or we'll cover some of the ones I didn't talk about as we go forward. I don't want to spend too much time, more than I already have, talking about that. Okay. I talked about cell membranes. I talked about that back in chapter 4. I just talked about it there. I want to just re-emphasize these are found in both types of organisms. They're a fundamental feature of all life forms. Here I just want to add a little more detail that I omitted in chapter 4. So when we look at cell membranes, this is again common to all life forms. There's really not a significant difference between a cell membrane of a eukaryote and a prokaryote. There are differences, but for our purposes of this class and this kind of level of biology, it's okay to think of them as being more or less the same. They in a lot of ways they're very, very similar, not identical, but as far as functionally speaking, almost the same. So a membrane is what separates the cell from the outside and the inside of the cell, and they're composed of a type of molecule, a specific type of lipid or fat called a phospholipid. So this is a very specific type of fat molecule. A phospholipid looks like this over here. Individually speaking, they look like this. You have two components to it. You have what's called a head and a tail. And the head is said to be polar, and the tail is actually two tails are said to be non-polar. The difference is that polar reacts with water, and it means it has a charge. In this case, it's usually a negative charge, but sometimes that can mean it's positive. So polar basically means it is slightly charged, either in a negative or positive sense. You can think of like a polarizing person is someone who kind of attracts attention to themselves or is somewhat charismatic. They're not neutral. They're not bland. They're kind of on one side or the other of being good or bad. So that's one way to think about it. Nonpolar is neutral. It means you have no charge. So if someone said you had a non-polarizing personality, that would be meaning that you're kind of bland and non-exciting, no charge to your energy. So in this case, we're looking at the tail as having no charge. So what you actually have in this molecule is two opposing charges, one that's actually charged and one that's not charged at all. And what this actually does is it generates an ability for the cell to create this boundary around itself with these opposing features. Now this is somewhat random because there's lots of ways you could create a membrane. So bear with me in the sense that this is just the way life works. There could be other ways, and I'm just kind of trying to explain it within the way that we actually see life working. So it's not to say that it couldn't have come about other ways, but the way it actually does work is that we see that we have the phospholipids. They arrange themselves in two layers. So we have what's called a phospholipid bilayer, and that's listed right here. This is just a very, very basic overview of a cell membrane. So a phospholipid bilayer means we have two layers of phospholipids. So here you can see an individual phospholipid, and we see the head and the tail. And on the other side, we see another phospholipid with the head and the tail, but they're actually pointing in opposite directions. So the heads on the phospholipid bilayer are going to face to the outside and the inside, and then the tails on both sides are going to point towards the middle. Now what this does is it creates in a scenario where the inside of the membrane, where all these tails are, they're actually said to be hydrophobic. And because they have no charge, what it ultimately means, and I'm skipping past lots of detail here, so bear with me, but what it ultimately means, what's important, is that means that that part of the membrane cannot interact with water in a way that water can stay within this area. So it means that this area basically is devoid of water, and that's what the term hydrophobic means. Again, I skipped a lot of reasons as to why. There's a lot of chemistry that you're going to have to know in order for that to really make sense. If you want to go look that up, I'd be more than welcome to do that. But in a nutshell, it means that there's a water-free zone, hydrophobic, meaning water-fearing. So this part of the cell basically creates a water-devoid area where it creates kind of this empty space that things generally don't go into. Most things interact with water, and so anything that interacts with water is not going to be found in this area. So you kind of have a dead zone. It's kind of like if you had a double-layered fence or a double-layered wall, this would be kind of like the space in between where you really wouldn't find too much happening. And the heads is that kind of like the bricks or the concrete itself. Now the head on each side is attracted to water. So what that means is that water can more or less come all the way up to the boundary of those heads, but it can't go past it, at least not in a conventional sense. There's more to that, but I don't want to get into too much detail. So water is basically stuck on one side of the cell and on the other side of the cell. We have a dead zone here in the middle. So what this does is it creates a very distinct boundary between the inside and the outside of the cell. And just one last little point here. Water is essential to life as we know it. So we have to have water in a cell, inside and typically outside to a certain extent. And so this creates a very efficient way to keep water inside and to allow water to surround the cell on the outside. If not pure water, some water-like solution like blood, for example, or some of your extracellular fluid, which is mostly water, your plasma, and mostly parts of your body that are mostly water. It could be some kind of body fluid. But we see that kind of distinct layering here. Now over here is a more advanced drawing of that. We see the phospholipid heads and tails. What we also see in a membrane are little channels and markers and lots of things embedded within and attached to, sticking out of the membrane. And those are some of the things that we're really not going to get into here. That's a topic for general biology. There's lots of little features. Well, I'll mention a few of those here in a second, but not lots of detail. And this is just another figure showing kind of a three-dimensional cross-section of a cell. So this is obviously a flat, very, very kind of basic two-dimensional. This is more of a three-dimensional showing the structure of the phospholipids there. So that's the idea of a membrane. Here's talking about the phospholipid heads. The polar heads face out, the non-polar tails face in, and that's what generates your boundary between the in and outside of the cell. So just a little bit about some of those things I mentioned embedded within the cell. You can think of phospholipids as like the base of the cell, or the base of the membrane of theirs. One of the things that really makes the membrane interactive are the proteins within it. So the proteins, I like to think of them as the quote-unquote gatekeepers, or you can think of them as like channels, kind of maybe like highways in and out of the cell. And what the proteins do is allow the cell to have an active and interactive component with the rest of the world around it, also with other cells within, say, a multicellular organism. Proteins, for example, are what, is a random example, what allow your heart to interact with your brain. Your brain is made up of membrane, phospholipid membrane. Your heart is made up of a phospholipid membrane. But without the proteins, there wouldn't be any way to actually connect those two things. They'd just be these inert little membranes. So proteins are kind of like the active and interactive components that make up the actual key aspects of a cell. So as some examples, what do proteins do? They can allow transportation in and out of the cell. That's where you can think of them as almost like gatekeepers or little highways in and out of the cell. They also allow for attachment. Go back here real quick looking at this. You can see these little structures here, like a glycoprotein, or these little branch-looking structures here. Some of these might actually allow the cell to grip onto something and actually latch on as an attachment feature. They also allow for communication and signaling. Going back here, some of these longer alpha helix type proteins can actually act as a way in which a molecule can land on one side and actually send a signal all the way through the membrane and have it reach the other side. Almost like a wire going from one house to another, almost like your phone lines. Remember when we used to use phone lines? It's kind of like that, where you can actually send a signal from one side of a house to another house, or from one house to another house, or one part of the country even to another through a physical wire. That's kind of like how communication might take place. Identification, another key feature. Some of these types of glycoproteins actually act as like the equivalent of a facial feature. The cells around them can actually recognize and characterize a cell based on these features that actually give it some kind of characterization to it. I like to think of these as like facial features. They allow cells to identify each other. It's actually something important. For now, we'll just leave it at that. But when we come back to I'll come back to this later when we talk about the immune system. The immune system is largely predicated on the ability of our body to recognize what we refer to as self, our own body, versus non-self, which is everything considered foreign. So our body is largely creating an immune reaction based on the determination of what's self and non-self. So that the idea is we want to attack things that are foreign and not attack our own body that's considered self. Well, the way that our body begins that process is by recognizing cell surface markers, these identification markers, and they can actually tell the difference between our own surface markers and those of foreign cells or foreign entities. And that's based on the unique characteristics of those cell surface markers. They are like a face where they're kind of similar, but each one is just a little bit different. And that's what allows the body to recognize self versus non-self. And then finally, some of the metabolic activities within a cell can actually take place within the membrane, and then others can take place within the cell. So those are some of the features of a membrane. Here's a figure showing some of those different activities. So transport, enzymatic activity, signal transduction like communication, a molecule hits on one side, passes a signal inside. Recognition, a feature here allows another cell to recognize. Attachment, adhesion, two cells can join together. And here's another type of attachment where a cell can attach to a different type of structure altogether. So some of those same things we just talked about there with a few illustrations for those. Okay, next we want to get into the eukaryotic organelles in a little more detail. As I mentioned, this is one of the two defining features of a cell, the internal organelles. And as we already mentioned, each is its own kind of independent functioning part of the cell, kind of like how organs have their own independent functioning within our own body as a whole. Now, this here is a figure of a eukaryotic cell right out of your textbook. This figure is meant to show all of the various features of a eukaryotic cell without saying that this is exactly what they look like. So This is not what all eukaryotic cells look like. In fact, probably most eukaryotic cells don't look like this. So don't take it as this is what they all look like, but rather this is a figure that we can look at to kind of get an idea of what they look like, and it's more or less to point out some of the different features that we'll talk about here as we go. If you remember from chapter 4, we had a similar feature or similar figure of a prokaryotic cell shown down here that was kind of the same thing, meant to give a generalized overview. The difference is that this one doesn't have any descriptions of the features like we saw in chapter 4, but similar overview figure. Now, the nucleus is one of the two defining features of a eukaryotic cell, but it's also actually considered an organelle. I know, go figure. The difference is that it's the most significant and most fundamentally characterizing organelle of the cell. So it's actually kind of specified in that definition as its own kind of thing, even though the truth is it's technically part of the organelle group, like other organelles, like the mitochondria, like the Golgi apparatus that we talked about earlier. So it's still an organelle, but it's kind of like the king organelle, and so they separated that out as a separate feature. And it's what houses the cell's genetic material, as I already mentioned. Here you can see some figures. These are both animations, and this is a real figure that's got some artificial coloring to it. But here you see some cells where you can actually see the membrane. This loose kind of boundary here is the membrane. And then they've basically just highlighted the internal nucleus to show how that would kind of look in the cell. And you can also see some of them are different size. Some of them are bigger, some of them are smaller. So they're not all the exact same size, but they do take up a fairly good chunk of a eukaryotic cell, and they're kind of one of the more prominent features of a eukaryotic cell. Here's another animation or 3D illustration of it. And this down here, I'm not sure if these are nerve cells. They kind of look like nerve cells. I'm not sure about that, but you can see these are real cells, and here you can see the nucleus taking up a big part of that. These are artificially colored, so the nucleus isn't normally blue like that. It's been stained to show, it's actually a fluorescent microscope image that makes it kind of stand out from the rest of the cell. So here it states that the primary purpose is to protect the DNA and also help regulate what goes in and out. Kind of like how our brain and our spinal cord kind of help protect the skull and the spine kind of help protect the brain and control what goes in and out. The nucleus is itself kind of one thing, but it's made up of three distinct components, at least three, and we're just going to focus on the three major components. One is actually called the envelope, the nuclear envelope, and that is what actually separates the nucleus from the rest of the cells. This is that kind of separate compartment, the separate covering that separates it, kind of like the skull of your brain. Nuclear pores are what allow things to come in and out, almost kind of like how the spine allows information to come in and out through nerve cells. And then the nucleolus is almost kind of like the major stuff of the nucleus, almost kind of like the brain tissue to a certain extent. It is actually what really makes up a lot of the core functions of the nucleus, and it's actually where ribosome synthesis takes place. So we said ribosomes are a part of what makes protein in the cell. Well, they themselves are created within the nucleus, so you kind of have a process of generating those within the key part of the cell. No, not too much detail there, just recognize those three parts and the basic idea of what each does. Okay, so next I want to talk about what's inside the nucleus in terms of the DNA. So as we mentioned earlier, DNA is found in the nucleus. It's kind of the key idea of the nucleus is to protect and regulate the DNA. So even though that wasn't mentioned on the last part, I want to talk about that now. I also want to talk about how it all fits in there. So the DNA is kind of its own separate topic, and we'll talk about that when it comes to Chapter 9 later in the semester, what it does, how it functions, and all of that. So for now, we're kind of talking about more of the structural parts of it. So that's kind of why I'm focusing on the idea of DNA compacting. So the idea is that we have all this DNA in our cells, and in a eukaryote, there is an especially large amount of DNA. So take a human being, for example. Humans have 46 chromosomes, and of those 46 chromosomes, if we were to stretch out the DNA of each chromosome end to end, we would find a total of about 6.5 feet of DNA per cell. Now, in your body, you have approximately probably 40 plus trillion cells. Those estimates vary. It all depends on how big you are. But roughly 40 trillion cells. And in every single one of them, minus your red blood cells, So that, I guess, maybe a slight exception there, but red blood cells don't have DNA within them, ironically. But despite, with the exception of those, every cell has approximately 6.5 feet of DNA in it. And they're so small that you could fit several thousand on the head of a pin. So kind of mind-boggling to think about that. So the only way that they get that much DNA within a cell is to tightly wrap it and package it and condense it into these really, really, really tight and highly organized bundles. And that's what we're talking about here. And this doesn't really happen in a prokaryotic cell. Prokaryotes have significantly less DNA and have a much less refined way of wrapping it. They kind of do condense it, but nowhere near on this level. So the way this works is we have specialized proteins called histones. Histones shown here are these little bead-like proteins. And what ends up happening is the DNA, which starts in a thread-like material, actually wraps around those histones, and then those histones kind of group together, as you see here, and then they wrap in tight and coil even further. And then ultimately these wrap into the structures that we know as chromosomes. So what you ultimately see is a wrapping and a folding and a condensing process that allows the cell to take 6.5 feet of DNA with, as an example in humans, and then condense that down until it all fits in a cell so small we can't see it with the naked eye. So that's how the DNA actually fits in there. So that's the last thing that kind of goes along with the structure. So within the nucleolus here, around the nucleolus, so between the membrane and the nucleolus, we would see all that DNA compacted in there. And they don't show it here because it would basically just fill every square inch of that nucleus. Okay, next part we're going to talk about is called the endoplasmic reticulum, abbreviated as the ER. Endoplasmic reticulum is what branches off from the nuclear envelope. And I mentioned that earlier in the drawing I made. It's a series of flattened membrane that is continuous with the nuclear envelope. So here you can see it in this figure. Here's the nuclear envelope. What ends up actually happening is it actually folds outward and starts to create a whole new structure that we call the endoplasmic reticulum. So it's almost like this extension of the envelope of the nucleus. And within it, what it ends up doing is forms a series of tunnels that you can see here that kind of connect and branch out from the nucleus itself. Now, as it branches outward, it actually differentiates into two distinct types. One is called rough endoplasmic reticulum. The other is called smooth endoplasmic reticulum. And so here you can see in this figure, on one side over here, we have rough endoplasmic reticulum, and over here we have smooth endoplasmic reticulum. The difference is that a rough endoplasmic reticulum has the ribosomes I mentioned earlier. Ribosomes are what give it a quote-unquote rough appearance. It's just a total arbitrary way of naming it. Somebody decided that's what they wanted to call it. It does make sense, but that's what they went with. Now, ribosomes, as we know, function in protein synthesis. And so what we see is that with those embedded in this part of the membrane, we end up with a structure that facilitates protein synthesis. So in other words, rough endoplasmic reticulum basically is a structure that organizes the ribosomes and collectively helps the cell produce proteins. So that's really what its major function is. It kind of supports the ribosomes, which then make the proteins. Now, in addition, it also helps transport those proteins, and it also helps kind of polish them to a certain extent. So there's kind of some fine touches that go onto them, and then it also helps kind of send those out to other parts of the cell, as we'll see here in a minute. So, but for now, just think rough endoplasmic reticulum, RER, think ribosomes, think protein synthesis. Now, other parts of the endoplasmic reticulum are referred to as smooth, SER, and they're considered smooth because they lack the ribosomes. So there's really a distinct difference in whether you have ribosomes rough or don't have them smooth. Now, the functions of the smooth endoplasmic reticulum are a little more complicated and not something that I want to go into too much detail here with, because you kind of open up a can of worms if you try and elaborate too much. You could really spend a lot of time focusing on that, because the functions are fairly diverse, and there's a lot of variation depending on what type of cell you're talking about. So, for example, your liver cells in humans, the SER is very different in terms of the exact functioning than it might be in, say, your brain cells or in the reproductive cells. there's very different functions. For example, in the liver, SCR has a more specific function of detoxification, whereas in, say, the reproductive cells, they function largely to make things like estrogen and testosterone. So, again, don't want to go into too much there, but they do a lot of processing, and this involves largely lipids and things like that, which explains some of the functions there. So for now, just recognize that they're smooth due to a lack of ribosomes, and that's really all the more I want to get into there. Just to clarify, though, from the study guide, when it says what are the functions of each, for the SCR, I'd like you just to know that they function in nutrient processing and synthesis, in storage of non-protein molecules such as lipids. Okay, so that line right there kind of tells you what's the function of the SCR. Be a pretty easy multiple choice type question. I won't go into too much more detail than that. Here's a picture showing the endoplasmic reticulum, a different picture. Here, this is out of our textbook, and this here is showing a close-up of the ribosomes embedded within there, kind of how they would be functioning. And then over here, you see an actual, what's called a transmission electron microscope image of the endoplasmic reticulum within a real cell. So here's the nucleus, and here's the different compartments of the endoplasmic reticulum. Okay, the next organelle is called the Golgi apparatus, and the Golgi apparatus is one that is often referred to as the quote-unquote UPS or post office, maybe even the FedEx of the cell. Now, one question I like to ask is why is it called this? And in fact, that's right off the study guide. Why is the Golgi apparatus often referred to it this way? The reason is because the Golgi apparatus largely functions to receive substances like proteins and lipids created from the endoplasmic reticulum. So it receives those products and then redirects them as to where they're supposed to go. So it says here, it's the site in which the cell, when proteins are modified and then sent to their final destination. Protein synthesis usually begins in the RER, the rough endoplasmic reticulum, and completes at the Golgi apparatus. So in the Golgi apparatus, what's happening is that proteins are being sent from the endoplasmic reticulum once they're produced into the Golgi apparatus. Now let me find a figure here of that. Here's a figure. So over here we see the endoplasmic reticulum. What ends up happening, I'll talk about how this happens here in just a second. What ends up happening is the products, like the proteins, are actually packaged up in these little things called transitional vesicles, and they're then sent across the cell and then are received by the Golgi apparatus, which is what you're looking at here. This yellow gold structure is the Golgi apparatus. So these proteins then are fused into the Golgi apparatus, and then they're literally modified and then repackaged as they come out the other side. So over here, they're actually transitioning out of the Golgi apparatus. And inside here, they're not shown here, but inside here, you would now have kind of modified and then repackaged products that are ready to be sent to wherever they're supposed to go within the cell. And on this next slide here, you can see a better overall figure of how that works. So here is the cell's nucleus. Here are the ribosomes within the endoplasmic reticulum. Here you're seeing some of these vesicles budding out. They would actually travel across the cell and then fuse with the Golgi apparatus. The products within would migrate through the Golgi apparatus and be modified, and then come out the other side and be repackaged, and then be ready to be sent somewhere within the cell. So what ends up happening then is that some of these vesicles will then migrate to the cell membrane, and then the products within here would actually be dumped outside the cell. So the Golgi apparatus, for example, might be involved in taking a protein that's made inside the cell and then secreting that outside the cell. An example of where that would happen in your body would be your neurotransmitters. Things like dopamine, serotonin, probably familiar with those. These are neurotransmitters that help our brain communicate. Well, those are proteins, and the proteins are made inside the cell, but they function outside the cell. So the way that they get outside the cell is through the Golgi apparatus. They're first made here, transported, and then sent to where it is they want to go. In this case, they want to be secreted outside the cell, and then they would go on to have an effect kind of in between two cells. You may be familiar with kind of how your nerves work. axons and dendrites and things like that. So these would kind of go from one nerve cell to another. In between there, would be outside of the cell, and they would help communicate. Now, also not shown in this figure, these vesicles can also be sent back inside the cell, and many of them will function internally. So the Golgi apparatus will kind of determine whether they go outside the cell for secretion or whether they're brought back in the cell for an internal processing. Okay, and that's not all that detail is not shown here, but that's kind of the idea. So this is what's referred to as nature's assembly line. The idea is that proteins, or rather the information in the cell, begins in the nucleus, extends into the endoplasmic reticulum, branches to the Golgi apparatus, and then ultimately is directed somewhere within the cell, either internally or externally. Let's just go back here real quick. That's why it's referred to as the UPS. Kind of like the UPS, you take them a package, they put a label on it, and that label determines where it goes. That's largely how the Golgi apparatus works. The proteins in this case are sent. The Golgi apparatus literally puts a biological label on them, and then that label is what directs them to wherever they're supposed to go. The only difference with the Golgi apparatus is that the Golgi apparatus actually modifies the package as it's being directed. Hopefully the UPS isn't modifying your package too much. So that's one difference. But in a sense that it receives, labels, and then sends, the Golgi apparatus can be thought of as the UPS. So that's a question I may just ask you in short answer. I don't know, but you might want to be ready to explain that in a couple sentences. Now, one more thing, this is also true of the SCR, just to go back here real quick. SCR, as we said, produces things like lipids. So some of the lipids are processed in the same way that proteins are. So these vesicles here could be branching off the smooth endoplasmic reticulum, in which case they would carry lipids as opposed to proteins. So really the Golgi apparatus could receive either proteins or lipids mostly, and then modify and redistribute those. Okay, hopefully that makes sense. If not, you might want to go back and look at that again. Always feel free to ask me questions. There are some really great videos on YouTube as well, and I try not to show those in here as much because I know it takes a lot more time. Now, one way that the cell actually sends things is through what are called cellular vesicles. Over here, they were referred to as transitional vesicles. It's kind of two ways of saying the same thing. I know that gets confusing. I apologize. The term vesicle is what's key here. The way that works is that you have membrane that is made of those phospholipids. So if we just go back here to refresh your memory, we showed phospholipids all the way back here. These phospholipids are very flexible and kind of squishy. It's almost like Play-Doh. Whereas if you were to squeeze this phospholipid, you can almost pinch part of that off and separate it. You could not almost, you literally, if you had microscopic fingers, you could literally grab a hold of that phospholipid and just take a piece of it off. Just rip it right off. And like Play-Doh, the rest of it would kind of fill in around it, and the cell would kind of reshape itself to compensate for that missing piece. It would literally kind of fill in the gap automatically. And then that piece could literally be pulled off. It would actually reform a new vesicle like this. It would actually go back into this kind of form where you've got the membrane heads on the inside and the outside. And you would literally create a separate little vesicle that was pinched off from the rest of the cell. So that's what's happening when we look at a cellular vesicle. What's happening here, this is a part of the membrane. Just go back here. Like this is what you're seeing, like right here. So here is this is part of a phospholipid membrane. So the phospholipid membrane is both on the inside and the outside of the cell. I didn't say that just to clarify. It's a slightly different construction, but similar type of material. So you can literally pinch part of that off, which is what's happening here. And then as it rips off entirely, here's one that's in transition, the membrane would just kind of reform and reseal itself. Within here, you would have some of the products being produced by the rough endoplasmic reticulum. So now those literally float across the cell and then refuse with the Golgi apparatus over here. And that's kind of what you're looking at here. So here's one budding off. You can see these little green specks have the products within them, like the protein. They physically move across the cell and then fuse with another component over here. And here's another figure from a side figure, a cross-sectional illustration showing the same thing. Products gather near the edge, it pinches out, floats across, refuses, and the products get merged into this secondary location over here. So this is how products get from the endoplasmic reticulum to the Golgi apparatus, by literally budding out, floating across, fusing, and then dumping those products into the Golgi apparatus. If you take it for what it is, it's relatively straightforward. If you think about it on a more in-depth level, this is pretty complicated stuff, and it's hard to explain exactly how that works, and we don't have the time to do all that. So, there's videos online that will give you a more in-depth overview of how this works. But for this class, you just kind of take it for what it is. These are basically little transitional membranes that carry products from the endoplasmic reticulum to the Golgi apparatus. And then, just to clarify, these are the same kind of things that would bud out from the Golgi apparatus and secrete things outside the cell or inside the cell. It's a little flexible membrane that can bud off and fuse on the other side of things. So the key thing here is that this is what is used to transport things throughout the cell. There's an example that I want you to be familiar with of a type of vesicle that goes on to have a more specific function. So there's more detail here than what we're going to cover, but I just want to add one little thing to it. So there's actually a certain type of vesicle that is modified in a way that gives it a special purpose, and these are called lysosomes. Lysosomes are a type of vesicle that contain digestive enzymes. So what they end up doing is being infused with a type of enzyme that will literally break down food-like substances into smaller pieces. Just like our stomach digests food, these are basically like an internal stomach of the cell. In fact, this is actually how your body digests things inside of its cells, which happens every day. So you can almost think of it like a little micro stomach for the cell. And what you're looking at here is a figure of how that works. You see the Golgi apparatus here, and you see these little lysosomes budding off of those, and they have these digestive enzymes inside them. As food particles are brought inside the cell, the lysosomes fuse with the food particle vesicles, and then those lysosomes go on to break down that food-like material. And then the energy from that can be redistributed throughout the cell, and then the waste can be eliminated through a process called exocytosis, where the vesicle buds outward and dumps that contents outside the cell. Here's another figure showing the same thing. Here's the food being brought in its own little vesicle. The lysosome merges or fuses with that, and inside there the food is digested by those enzymes. So that's just one example of a handful that we could talk about of a cellular vesicle that has a function of digestion. Also, we'll talk about this later in chapter 14. We'll talk about how this is modified in our white blood cells to actually be a component of digesting invading microorganisms. So we talked a little bit about phagocytes in chapter 4. Phagocytes are your white blood cells that digest and kill microorganisms. Well, one way that they actually kill the microorganisms is by using lysosomes. So in some cells, this is a food that's digested. In other cells, it's actually an invading microorganisms like bacteria and fungi and algae and things like that. Or not algae, but yeast is what I meant to say. So I know this is confusing. I know it's a lot to take in, but this is why I have the study guide. Stick to what it is. The questions are asking. So what is the function of a lysosome? It is to digest, and it is basically a special type of cellular vesicle. Okay. Here's a quick video of the lysosomes.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
Okay, so they're pointing out another feature I didn't mention, which is how these can actually be used for internal housekeeping, so to speak. break down worn out organelles and allow the cell to generate new ones to replace them. Okay, a couple more organelles and then we'll move on to some different topics. There are what are called energy generating organelles, and there are two specific types here. One I mentioned earlier is the mitochondria, the powerhouse of the cell. This organelle shown over here, this is a real microscope image with the orange coloring, and this obviously here is an illustration. But the mitochondria, fairly complex little thing. I don't want to get into too much detail. It's another can of worms you open up if you try and talk about it too much. So really keep it very simple, and that is in the sense that the mitochondria is an energy generating organelle, and then what I want you to know is it generates ATP from glucose. So glucose is a sugar. Your body converts, obviously sugar can be used, I should say sugar is used directly to make ATP, but your body can even break things down like fats and proteins into sugar, which can then be used by mitochondria. So there's a lot of ways to create glucose in your body through a variety of food sources, and your body does that and then ultimately feeds the mitochondria with that glucose to make ATP. It can use other things besides glucose, but that's kind of its main energy source. So that's the mitochondria. If you want to know more, there's an interesting connection in evolution called the endosymbiotic theory, and there's just not enough time to talk about all this, but for anybody who's interested in evolution of eukaryotes, the endosymbiotic theory is a proposed mechanism by which eukaryotic cells evolved from prokaryotic cells. And interestingly, what it suggests, with a really high degree of certainty and lots of evidence to support this, what it basically suggests is that mitochondria are actually bacteria that emerged, or co-evolved rather, with the eukaryotic cell to create a new source of energy for the cell. So it's kind of an example of co-evolution between two organisms that went on to become one organism, and we actually see that the mitochondria looks and has all the features of a bacteria. So the theory is pretty sound in that this thing has all the features of a bacteria. It's just that it's no longer a separate functioning organism. It's now part of a eukaryotic cell, almost like a slave to the eukaryotic cell. And its main purpose is to generate extra energy. The same is actually true of the chloroplasts. These are both thought to be bacteria-like organisms that were kind of trapped within eukaryotic cells with the purpose of generating energy. The chloroplast is a different type of organelle in that it generates, or rather it conducts photosynthesis. So it's a different type of energy generating process. So photosynthesis is unique to plants and algae. There's also bacteria that do this, but from a eukaryotic standpoint, these are both plants and algae. Remember, I compared those two together back in chapter 1. Don't really want to say too much more, but just recognize that these are the two energy generating organelles. Mitochondria makes ATP from glucose. Chloroplast generates energy through photosynthesis, which, if you're not familiar with that, which is where light, water, and CO2 generate glucose. And that's actually how glucose is formed on Earth. So, okay, moving on here. Ribosomes we mentioned in chapter 4. Talk about those again here. Remember, those are similar features in both types of cells. Ribosomes, protein synthesizers, we've already talked about that. This shows you a better structure, a better figure of the structure. Ribosomes are actually made up of two specific units. One's called a large subunit, the other's called a small. And they actually function when the two come together and almost sandwich together, kind of like hamburger buns. I always think of the ribosome as kind of like a hamburger bun. The only thing I really want to point out here is that while both cell types have ribosomes, there is a difference between the exact types that they are. So eukaryotic ribosomes are said to be what are called ADS ribosomes, kind of an arbitrary name that they just kind of made up based on some of the properties that they observed in the lab. So I don't want to get into why they call it that, but just recognize it's called the ADS ribosome. And prokaryotic ribosomes are called the 70S ribosome. So while they both have ribosomes, they are two distinctly different types. I like to compare them to two different types of trucks, like a Chevy versus a Ford. Chevys and Fords are both trucks. They both have four wheels. They both have a truck bed. They both have an engine. They both do the same thing, but structurally they have different components, and if you take them apart and tinker with the internal gears, They're just a little bit different, and that's kind of how ribosomes are. Do the same thing, largely look the same, but there are different structural components to them. This is only significant because one of the things we'll talk about later in this semester is how these types of subtle differences can lead to significant ways of targeting one type of organism or another. For example, we can use antibiotics that actually target the 70S ribosome and not the 80S ribosome. So drugs like tetracycline are actually specific to only targeting the 70S. It would almost be like having a virus that targeted the computer board of a Chevy truck and not the computer board of a Ford truck. You know, all trucks and cars have these computer chips built within them, right? So imagine a computer virus that you could insert into the motherboard of a Chevy, but not that of a Ford. It's kind of like how antibiotics work. It's a drug that only targets one part of the 70S in this example and doesn't affect the 80S whatsoever. So when you take a drug like tetracycline, you have this ability to target only the 70S and not the 80S. Okay, the next thing I want to talk about is what's called the eukaryotic cell cytoskeleton. The cytoskeleton is essentially a group of proteins that help kind of make up some of what I like to think of like the scaffolding and like the structural beams almost of the cell. So you can almost think about like a large building, and you have to have support beams, and you have to have reinforcement around the outside of the building. You have to have kind of this architectural design that allows the building to be supported on a large scale. So just to give you a few visuals, think about it like this. Here's some pictures of a building. You've got some of these cross beams, and you've got these designs that are meant to kind of help reinforce the shape of the building. Ideally, it helps it not collapse from within. You've got these key designs in beams and angles to kind of help redistribute the weight, kind of help make up some of the ability of these buildings to hold extra weight. Also, what allows the building to get larger. You know, you can't make a really, really large, complex building without a key set of designs that kind of allow that building to hold extra weight and support itself. So in a eukaryotic cell, you can think of it kind of the same way. Remember, eukaryotic cells are much bigger than a prokaryotic cell. And one of the things that happens as you get bigger is you have to get more structurally reinforced. So we don't really see this in the prokaryotes, and that has partly to do with why they're small and also how they can get away with being small. So if you make a really tiny little, almost like living in a tent, for example, or like a really small little shack, you don't necessarily have to spend as much time on the key structural reinforcement of that building. If you want to make a really, really large house or really big complex design in reference to a building or architecture, again, you have to spend more time setting up the support beams and kind of how all the weight is redistributed and how many people you can hold and that sort of thing. So that's kind of like what we're looking at here with the eukaryotic cell cytoskeleton. It's really kind of like an architectural design of the cell that allows it to be supported. It also goes on to do other things such as help with movement and transportation within the cell. So it goes a little bit beyond that. So it helps to anchor organelles. It helps to transport cellular vesicles. It helps to permit shape changes and movement. And that all is kind of part of within this idea of kind of creating the scaffolding and the structural support of the cell. Here you're looking at a figure out of the text. This is showing that figure we showed in the very beginning, the overview figure. And you're seeing here these little hair-like structures. These are all part of the cytoskeleton. This here and down here, these are fluorescent microscope images of cells in which they've highlighted just the cytoskeleton. So you don't see the nucleus, or you don't see rather the cell membrane, you don't see the other organelles. You see just the cell cytoskeleton, plus in this case you actually see the nucleus. So this blue is the nucleus. You kind of see where the center of the cell is. And then all around it here, you see just the cell cytoskeleton. So you can see how this kind of permeates inside the cell and generates this kind of scaffolding that the cell can then kind of be supported around. Now, there are three types. So just real quick, you want to know what are the three functions. You also want to know what are the three types. And each one is a little different. So you also want to know just a basic idea of what each one does. I'm not going to ask you real complicated, in-depth questions on this. I just want you to have a basic understanding of the three functions, the three types, and a basic idea of what each type does. So one type is called a microfilament. Another type is called a microtubule. Another type is called an intermediate filament. Each one is a little different. Microfilaments are these long, kind of thread-like proteins, and these are found in the highest concentration around the cell membrane. So going back here, you can see the microfilaments. By the way, this is where biology gets frustrating, I know. They also refer to these as actin filaments. So actin filaments and microfilaments are the same thing. I apologize, I didn't have that stated here. But what they do is make up a lot of the scaffolding around the outside of the cell. Let's see here. You can see the microfilaments, called microfilaments here, and you can see here's the membrane in blue, and the microfilament is attached directly to that, meaning it makes up just around the outside of that cell. One of the big things that they do is they help the cell in reference to movement and contraction. Okay, real quick, I found this a part of a video I want to show you that talks about how microfilaments can actually be responsible for cell movement and contraction. I have to mute the sound because it's absolutely horrible quality and a very poor explanation. But let me just kind of walk you through just a real quick video on mute to show you kind of what's happening here. So here's a cell. This could be a eukaryotic cell of almost any kind. What we're going to see here is how the actin filaments, and they're called actin filaments in this video, again, same thing as microfilaments. We're going to show how they work. So what's going to happen here is that the microfilaments, the actin filaments, are going to push the membrane. So this is the membrane around the outside of a cell. We're going to push the membrane in one direction, and what that's actually going to do is actually move the cell in that direction. So it's going to kind of push it in one direction by the actin filament extension, and then we're going to see the back half of the cell kind of contract, and what we end up seeing is what's called cell crawling, in which the cell is pushed in one direction and then kind of contracted, ultimately kind of gliding it in one direction. So here you're seeing a part of the cell that's going to be involved in movement, and we're going to see here how the actin starts to actually concentrate. I'm not showing here. Here's the actin. Here's the filaments that make up around the outside of the cell. What we're going to see is that they're actually going to start to grow and extend and actually physically push the membrane with them. So here it's showing there's the actin growing, and what that actually does is it physically moves that membrane forward. So you're actually seeing movement take place in real time through the extension of the actin filaments or the microfilaments. So as they push forward, that actually causes the cell to move. So here's another view of that from the side. So this is like a single-celled amoeba type organism. And right here in red, this is where those actin filaments are extending, and it's going to push the organism in this direction. So it talks about how it anchors here. That's a little bit extra information. But here it shows that extending outwards. That's through the microfilaments or the actin filaments being pushed out. That physically moves the organism forward just a little bit. And then what ends up happening is some of the other actin filaments here inside are actually going to contract themselves and actually bend forward. So you kind of see an extension followed by a contraction on the back half. And this is done very, very quickly. This is obviously shown a very slow moving time here. So here's another part of that contraction. So this is all taking place in real slow time here, but organisms do this very rapidly, and they extend out one part of the cell, and then they contract the back half of that cell. Real quick here, this is when I pull up. You can see this in real time here. This is a real organism. So you can see how this would be extending forward and then contracting on the back half here. Hard to see all that. Life is really complicated, and this is taking place really fast. But that's actually what's happening on the microscopic level. You're actually seeing, this is kind of from the same source here, you're actually seeing those extensions followed by contractions that are taking place in real time. So that's actually one way that certain cells can go about moving, by using part of the cell's cytoskeleton. That's a pretty amazing thing that they do, which is to extend and contract, and that actually leads to cellular movement, which is usually referred to as cell crawling. Not all cells do that, but for a lot of cells that move, that's one way that they can go about doing that. Now, another key part of these cytoskeletal features is called the microtubules. These are long, hollow protein tubes, and those are shown here. Let's find just a slightly better figure of those here. go. Usually they show, here's a better one. Here we go. There's kind of a microtubule. You can see it has a hollow structure. Here's a cross-sectional view made up of a couple different proteins called tubulins. So these literally create almost like a PVC pipe type structure, which is hollow on the inside, and that then allows it to serve its function, which a lot of times is part of the overall shape, but it helps to transport substances and allows for movement of things in and out of the cell. It also is a part of the flagella and the cilia in eukaryotic cells that we're going to talk about next. So it actually helps to create a new structural complexity of the flagella that helps it move back and forth. So we'll see that here in a second. This is a really cool video, totally animated and a little bit dramatized. But just to give you an idea of how microtubules might actually be responsible for movement, and also actually how vesicles move throughout the cell, I want to show you this video. So this is a microtubule, and this is a cellular vesicle, one that we talked about earlier. So earlier I just mentioned how these just float across the cell, kind of like I showed here. So here's the vesicle, and we said they just kind of float from one side of the cell to the other. The truth is, they don't necessarily do this so magically. They're actually being drug along microtubules. So we just won't always show that detail because it's so complicated. You can't necessarily just put every piece of detail in every drawing and every figure. So to give a little more detail here and give you a better idea how that works, you actually have a microtubule that would extend throughout the cell, and here's the vesicle, and the way it moves is by these little proteins called kinesins, and what the proteins actually do is move along the microtubule. And they literally do so by activating these two little leg-like structures that physically carry that vesicle from one side of the cell to the other. This is real, by the way. This is not fiction. This is a real thing. This is actually what's called a motor protein. And the motor protein is being activated by energy, and it literally drags that vesicle from one side of the cell to the other. Very short little video there, but that's actually how things typically move about the cell. So when we talk about how it's responsible for cellular movement, really what it does is it acts almost like a little road by which vesicles can travel along with the assistance of other little proteins, in that case a motor protein. Life is really complex, and so that's just a kind of a little more detailed glimpse into how all that actually kind of works. The last type is called an intermediate filament. These are often found throughout the cytoplasm and often help reinforce some of the organelles and things like that. So mitochondria, for example, here are shown. This is a mitochondria, and you can see some of these intermediate filaments here kind of helping to make up the internal scaffolding and kind of help reinforce where these organelles go. Also might help kind of reshape some of the organelles throughout the cell. Okay. Next thing, we want to talk a little bit about the motility appendages for eukaryotes in terms of their extensions from the cell. In chapter 4, we talked about the flagella and the fimbriae. We also talked about, I misspoke, just the flagella for motility. Bacteria have only flagella for a motility feature. In eukaryotes, we see a very similar structure. Actually, it's very different structurally, but similar functional, I should say. It's actually called the same thing. So we have a tail-like structure here that is very similar to that of a prokaryote in function. So there's a couple different types here. Here's a tail. Here's a series of tails, all flagella. So it's similar in function. Similarities in that it's used for movement, extends out, and it moves in response to stimulation. We didn't really talk about that in chapter 4, but just to clarify, the tail is moved based on some kind of stimulation. They don't do so randomly. They wait till they have either a positive or a favorable stimulation, and then they either move towards it or away from it. So all that is similar. The key differences here is that it's significantly more complex. The eukaryotic flagella is said to have what's called a 9+2 arrangement, which gives it a more dynamic range of motion and makes it act like a true tail. We talked about the prokaryotic flagella, and I mentioned that it's very, very simple. It really kind of only moves in two directions. It either can rotate clockwise or counterclockwise, and it really only has a couple basic lines of movement. It can either kind of move straight or it can kind of move randomly and reorientate. So it's very, very simple in terms of its actual ability to direct a cell with a certain level of precision. Bacteria kind of move clunky and random, and it takes them a long time to get anywhere. The eukaryotic flagella is much more complex. So this is a side view of the flagella extending out from the membrane. When you look at it inside, it's actually got these structures here, which are the microtubules, shown in a series, which is what we refer to as the 9+2. So here it's another figure showing the microtubules, what's called central and double microtubules. What you essentially have are 9 pairs of microtubules around the outside, or rather around the periphery of the flagella. That's where the term 9 comes in. And then you have 2 microtubules inside here paired together, and that's where the 2 comes in. So when we say it has a 9+2 arrangement, that is in reference to this organization here with 9 and then 2 in the middle. Again, very random name. They could have called it anything, but that's what they decided to call it. Now, the way this works, these microtubules will actually kind of glide past each other and allow the tail to move in a variety of directions. Okay, real quick, let's take a look at a real quick animation here on mute. This is going to show kind of how the flagella begins, how it extends, and then kind of how it moves up and down. So here we're looking at the beginning of the flagella formation. These are structures called centrioles. They're going to divide, and they're ultimately going to move towards the membrane. Now, these have multiple functions. Centrioles are used in cell division also, but here they serve towards the production of the flagella. So they anchor into the membrane, which now they're called a basal body, and they literally kind of extend outward. And so this becomes the beginning of a flagella, also cilia, which we'll talk about next. So here you're looking at the 9+2 arrangement. Here's the 9, here's the 2 in the middle. Now what you have here, these little white structures are called motor proteins. What can happen is the motor proteins can actually slide down past each other on one side or the other. So at first they're nice and parallel, but what can happen is on one side of the flagella, the motor proteins slide down. What that does is it causes tension, which makes the flagella move like this. You're basically pulling this side like that, okay? Or you can pull the other side like that. And you can do this in any direction. So this is where you can get really all 3D dynamic motion. So when one side pulls, the flagella bends in that direction or the other direction or forward or backward. And that's how the flagella or the cilia are going to move. So here you can see that. Got it on mute. And it's going to start moving here in a second. Here we go. So here you can see as it starts to bend and move. So in this case, the flagella are bending down and then they're bending up, and that's all based on the motor proteins that are sliding past each other. And yes, you can come up with all sorts of jokes right now, and I'm sure that's really funny, but that's the idea. So you can get movement based on the sliding of these proteins back and forth, and that can happen in any direction, which gives it a very, very dynamic, three-dimensional sense of rotation. That may not make perfect sense, but hopefully it makes a little bit of sense. So really all I want you to know is the 9+2 arrangement and kind of how that gives us a more complex variety of motion. Now, we're not going to go through all of the different arrangements here like we did with the prokaryotes, because there's so many of them that it would take us a much longer amount of time. So there's a huge amount of variation as to how these flagella are arranged, and so we're just going to skip past that. Now, there's another similar structure called a cilia, which is a smaller version of a flagella and typically more numerous. So just go back here real quick. Here's a flagella, kind of a traditional single tail. Here are cilia. They're the same 9+2 arrangement, but they're just shorter and more numerous and stick out all around the outside of the cell typically, although I'll talk about some exceptions to that. So I like to think of flagella as more like one major or maybe a couple major motors, that is, on the back of a boat. Think of like a flagella like this. Think of this as like a little boat in the water. Think of this as like one big motor on the back, and you kind of have one big mechanism of propulsion. cilia, I like to think of like a Viking ship with lots of little oars on both sides. And here's a visual for you there. So kind of like how you have to have lots of little tiny oars all moving simultaneously. That's kind of how the cilia work. They all kind of beat in unison and kind of propel the organism forward, kind of like that. Whereas the flagella, again, is more like one big propulsion device at the back. Now, so cilia are used for movement. They're also used in some organisms for filter feeding, and these are referred to as oral cilia. So here's some figures of different ciliated organisms. Here you see cilia all around the outside. Over here, though, I know these aren't the best drawings. I'll get a better one here in a second. Over here, you're seeing cilia just around the mouth or the oral cavity of one of these organisms. And what they can actually do is kind of draw food inward to their cells. So here's a better figure of an oral cilia here. So this is an organism that would anchor itself to the bottom of an aquarium. And here you can see these little oral cilia that are around the opening. And what they do is they kind of flap and they actually draw nutrients from the water towards them, and then actually they stick to the cilia and then they can be absorbed for internal digestion. So that's another way that cilia can be used. Some organisms use them for one or the other. Sometimes they use them for both. So here you see some other illustration. These are actually all microscope images. These are all real images of ciliated eukaryotic organisms here. Okay, so basically they're just smaller flagella that are more numerous, and they serve, in addition to movement, also a function for filter feeding. Last but not least, we have in eukaryotic cells what can be referred to as cell connections. And there are a couple ways that they work, but they all kind of help to join cells together, which leads to the formation of multicellular organisms, but really it leads to what we refer to as tissue. So when we look at how multicellular organisms come about, they go through the process of individual cells joining together to make tissue, and then those going on to make organs and organ systems. Let's just show your cell, tissue, organs. There's a general flow here. So for example, you can see cells group together to form tissues, and then tissues go on to make organs and organ systems and an organism. But right now we're looking at how cells group together to form tissue. And the idea is they have to kind of stick together and attach together to form kind of a group of like cells. Here's another. I guess I lost it. Oh, here it is. This is a microscope image of some plant tissue. You see lots of cells all kind of joined together. So the way that they do this is with these cell connections, and they literally tie themselves, so to speak, together to form this clustering that we refer to ultimately as tissue. So there's a couple ways this works. One is called an adhesion junction, and this connects cells in a strong but flexible nature. So here they call it an adhering junction. And basically just between two cells, this is a way for them to kind of stick together. Another type is, so this is kind of a basic connection mechanism there. Another type is a little different in that it's called a gap junction, or it's different in the sense that it allows things to travel back and forth, I should say. So these are called gap junctions. And what they do is they allow little tunnels in between two cells. And this is actually how substances can move from one cell to another within tissue. So this allows cells to kind of all have this kind of intricate connection system in which substances and nutrients can flow from one cell to the next. So that's called a gap junction. They allow direct cell-to-cell communication, as well as a variety of other things like nutrient sharing and things like that. The last type are called tight junctions. Tight junctions are kind of similar to adhering junctions, but the difference is they literally create a watertight layer between two cells. So anytime two cells want to mesh together in a way where nothing can really move between them, tight junctions are found. These are found, for example, in your intestinal lining, especially around the stomach, where you have gastric juice or gastric acid, like hydrochloric acid in your stomach. And this allows your cells in the stomach lining to be so tightly wound together that your stomach acid can't just easily leak out into the rest of your body. So tight junctions are similar but form a very, very tight waterproof layer between two cells. An example would be like in your stomach to keep the stomach acid from leaking out. oh, here was that same picture. Look at that. I forgot I had it there. But anyway, this is ultimately the first step in going from a single-celled type organism to a multi-celled organism. And it ultimately begins as one cell groups together to form a tissue, and that is based on these abilities to form junctions between each other. Okay, so that's kind of how that would lead to a more complex level of organization. Now, that's where you get into anatomy and physiology, and it kind of takes over from there to explain all that, the rest of that detail there. But from a microbiology standpoint, we leave it there, and we conclude here with Chapter 5, Part 1. Okay, we'll go on for Chapter 5, Part 2 here next, and as always, feel free to email me with any questions, and we'll take a look at the next part here in just a second.